it all started with a patient who was admitted to the Montreal Neurological Hospital at a time when Jean Aicardi was visiting. Jean Aicardi has been one of the pioneers of the identification of this disorder, quite a rare disorder. And it started because he happened to be visiting at the time when this boy was in the hospital. This patient was in the hospital. It was actually a girl. And so this eventually led to further discussions, and then finally to the publication of a monograph on alternating hemiplegia of childhood, of which the three editors were, in addition to myself, Jean Aicardi from Paris and Federico Vigevano from Rome, also a distinguished pediatric neurologist who had experience with the disorder. So that's how it all began. And then eventually the American and Canadian people became involved in the association, which met here a couple of days ago. And this has led to, finally, after a period of relative stagnation, it's led to quite a lot of progress. And that, I think, has been quite interesting and very promising as far as better understanding and progress towards trying to understand the mechanism of this disorder, which still remains mysterious, but the progress has been very encouraging. Eva, what do you think?
Yes, I agree. So I'm Dr. Eva Andermann, and I do neurogenetics at the Montreal Neurological Hospital and Institute. And I have been involved in this disease almost as long as my husband, because he referred the patients and families to me, hoping that there might be a genetic explanation of the disease. And since all of the cases that we saw were sporadic, that is, there was no known family history, at that time we were not even sure that it was genetic, because there were no diseases, there were very few diseases at that time, especially diseases like this with paroxysmal disorders, which were thought to be metabolic, where you would have a new mutation. Most of these diseases were autosomal recessive. So if there were only one affected in the family, you might think, well, especially if the family, if the number of siblings were not great, that just by chance the others were not affected, but that this was probably a recessive disorder where both parents were carriers of an abnormal gene, but carriers were perfectly healthy, and it's only when the child inherited one abnormal gene from each parent that they would have the disease. So that was the hypothesis at that time. And it was actually, well, as you know, there was just one family where it seemed to be an autosomal dominant disease where there were several generations affected. And that suggested a dominant inheritance rather than recessive. But in all the other cases to date, there have been only single cases or sporadic cases. And the finding has been that these are new mutations, because the parents have been tested and they were not found to be affected. So only a couple of years ago, when the gene was discovered, that we were able to ascertain that this is the mechanism, which is a rather rare mechanism, that all of the patients were due to new mutations. And I think that this was probably discovered just now, well, for two reasons. That the technology is now available to look at the entire exome and to do what we used to do in 2 years, now can be done in 2 days. But then the analysis and the interpretation are not that easy. So, as I say, I got involved in the 80s when my husband first became interested, or perhaps even earlier, in this disease. And I saw many families. I did detailed family histories, and indeed we did not find any evidence that other family members were affected or had even any of the symptoms of the disease. And so for a long time, as my husband said, there was really no progress because we were not able to sequence the entire genome until, as you know, just over 10 years ago. So as far as the genetic aspects, we really didn't make much progress. We tried, there was only this one family that several groups studied, including Dr. Swoboda's group and Dr. Mikati, and they could do linkage studies on that family, although it wasn't such a large family, so they couldn't be sure exactly, exactly the location of the gene. But then, until the new technology came around, they really couldn't identify the gene. And we had this meeting in Rome, first of all, many years ago, and that was the basis for the monograph that my husband and Dr. Aicardi edited. And at that time, the genetic background of this disease was really not known. And then so it was a period of almost 25 years from the time that we started looking very carefully for a genetic cause until we got to where we are now. Now, what was discussed today and what is really necessary in the future is to continue the study of the genotype-phenotype correlations, such as was done by Dr. Sasaki, so that the specific mutations can be characterized as to what the phenotype or the clinical picture that they produce. And then we have to deal with the patients who so far do not have a genetic diagnosis and do not have a mutation in the ATP1A3 gene. And even though of the classical cases. This is only a small proportion, about 15%. There are many other phenotypes which are similar but not the same, and the gene for the moment is not known. So we discussed today many of the future research possibilities to try to identify the gene defect in these patients, either to try and still find a mutation in the same gene or to look for mutations in other genes. And this is what still has to be done from the genetic point of view in the future. And also, very importantly, because this is such an unusual finding of the new mutations, it's very important for genetic counseling for the families. And since it's not the common thing that is usually found, it's important that the genetic counselors can convey exactly what the risks might be and learn about this form of inheritance. Well, in terms of the natural history and the prognosis, in most cases, although not all, it is quite severe, which is surprising because in other conditions similar, where you have hemiplegia, for example, hemiplegic migraine, the results are definitely not as severe as this, and usually the patients recover entirely. So from that point of view, it's an important disease to pursue in order to try to prevent the bad outcomes of this disease. From the genetic point of view, as I mentioned, this is only being found now in several diseases, for example, Dravet syndrome, which has exactly the same thing. There are just a few families where more than one individual is affected, and most of them are sporadic, and yet it is due to a genetic cause. The same thing, it is due to a new mutation in a sodium channel, which was just not envisaged before, and almost all the patients now have been found, between 80 to 90%, to have these new mutations in the same gene. And until actually Dravet syndrome, I'm not sure that there were any diseases of this nature which seem to be either metabolic or because certain diseases such as malformations, which were static, were due to new mutations. But these diseases such as Dravet syndrome and such as alternating hemiplegia, one would never have thought that this was the mechanism, the genetic mechanism for it. So it is something new that we have to learn, and we have to learn how to counsel the families accordingly.
But the major problem that remains is to understand what the mechanism of the disorder is. And even though there has been this very striking progress in the understanding of the genetics of the disorder, the mechanism remains obscure. And I think that one has to walk before one can run. And the first attempts should be to try to identify why this mutation, produces this kind of clinical picture. And then one can address the issue of how the disorder can be treated. And how long this will take is hard to be sure. But certainly for us, and I think for the families who are here, and for the other people working in the field, it's been very satisfying to see the progress that has been made in the last few years, which is, I think, a major step forward in our understanding of the process and in our understanding of the disorder. And of course, everybody, not only the parents, but the people working in the field, are very hopeful and anxious to try to find a mechanism. And I hope that this is going to be accomplished in the not too distant future. But it's very difficult to predict exactly what this mechanism is going to be. If it were easier, that would have been done quite a while ago. Well, the treatment is still problematic. When you look at the list of drugs or agents which have been used or have been tried for the treatment of alternating hemiplegia in an attempt to change the outlook and to change the future, to change the prognosis, they vary enormously. And I think that it's very hard to identify a treatment which is going to work optimally, which is going to work all the time in everybody and completely. And so what we see here is that there are a number of agents which seem to be helpful, but they do not really result in a complete cessation and complete disappearance of the mechanisms of the disorder. And flunarizine, which is a calcium channel blocker, has been used, and I think it was Paul Casser, the Belgian neurologist and molecular scientist, who identified this agent, which has been actually produced in Belgium, if I'm not mistaken, as a treatment of the condition. And it certainly does bring about improvement. It helps, but it doesn't really remove all the problems that people who have this disorder have. And so that it remains a mainstay of attempts to treat the disorder. This drug, flunarizine, doesn't seem to have very much economic potential. It doesn't seem to produce very much income for the drug company that developed it. And so that they haven't really tried very hard to bring it on the market in different parts of the world. And as a consequence of this, it's often difficult in many countries to obtain a supply of flunarizine for the families who have an affected child. And that's where we stand at the moment. I think the important thing is to try to accept that, first of all, one has to find more about the mechanism of this mutation. And then I think it's going to be much easier to try to find modifications which can be brought about to the genetic makeup, which may hopefully control the disorder more fully and enable the children who have this disorder to lead a more normal or a normal life, which is the hope that not only the families but all the people involved, including the makers of this video, have in mind.
What I was referring to is to sequence the entire genome, for example, and to find the gene. Now that a gene has been found, as far as the mutations are concerned, that is already available for 85% of patients. And the next steps are, what is the function of this gene? And these studies are now going on, and this was discussed at the meeting here yesterday. And the functional studies are going to be very useful to give us clues what might be given to these patients to counteract what the mutation changes in the protein. And sometimes these are small molecules that can be given, because since this is a brain disease, it has to go through the blood-brain barrier, and that is a big problem for many drugs. And as you have heard yesterday also, one way is just to scan drug libraries and look at cells of patients in vitro, or look at animal models and give them hundreds, thousands of different drugs, which may be used for other diseases which have been approved, and therefore that would be a real shortcut. If you could find a drug that's already been approved for another disease where you don't have to go through as many studies as are required, as was said yesterday, that could take 15 years just to do the development of one drug. So if you have one that's already been developed and approved by the FDA and other agencies for other purposes, and if that drug happens to work, happens to work in cells with AHC, or of patients with AHC, or in animal models, then the next step would be just to do the clinical trials in patients with AHC without having to do all the early developmental steps, because the drug has already been tested for safety in humans and so on. So that's one way. So one way is just to do sort of a random fishing expedition using thousands of drugs. And the other way is to go by the mechanisms or the functional studies that are being done with specific mutations to see what effect they're having on the protein, and then to try to counteract these effects. For example, if there's problems with the folding of the protein, and you find another protein that can correct this. And so these are the two ways. Either you just try by trial and error, or you go by the specific mechanism.
Now, it's clear that this gene that was identified represents a major step forward, but it's probably not the whole story, because there are a number of patients who don't seem to have this gene, and yet they have pretty convincing, the disorder that they have is pretty convincing that it's very, very similar to the disorder that the people who harbor the gene have, all the way up to an autosomal dominant, that is a transmitted from generation to generation problem of alternating hemiplegia. But unlike what you find in the sporadic form, some of the families where this is transmitted from one generation to another only have attacks which start during sleep. They don't start in the waking state. And not only do they start during sleep, but the people who have this do not show any evidence of loss of intelligence or loss of ability, and they remain just as inconvenienced by the attacks, but at the same time they don't lose any of their faculties. So here you have an example of at least one condition which is analogous or similar, but clearly not the same. And of course, amongst the people who do not carry this gene, there may be several other forms, several other mechanisms of action which we can only think about, but which cannot really be proven so far. So that a great deal of clarification is still required in order to understand the disorder better, and then eventually there will be, as my wife has pointed out, attempts at more rational treatment of this process, which I might add has been, according to the parents and according to the people who witnessed this, it's been a pretty terrible disorder.
Well, just the last condition that my husband was talking about, and we've actually described this as benign nocturnal alternating hemiplegia. And this is probably a different disorder, at least genetically. Clinically, it's similar, except that it starts during sleep, whereas alternating hemiplegia usually starts during the waking period and is stopped by sleep. Interestingly, that condition also is relieved by sleep, but it actually starts in sleep, and then they wake up, and then it's again relieved by sleep, isn't it?
That's correct.
So here we really thought that there is more likely to be an autosomal recessive condition, because we have seen it in siblings. But this is not yet known, and as a matter of fact, I'm trying to lead a study to find the gene for this disorder.
For the nocturnal...
For the benign nocturnal alternating hemiplegia, and any clinicians that have patients with this, I would be very interested in. And so far, we wrote up about 5 or 6 families with this, and and there have there've been maybe another 2 or 3 in the literature. So it's very rare. But interestingly, the very first paper in 1971 of Verrett and Steele, they included 2 brothers who had the nocturnal form in their paper. So, but they didn't sort of differentiate it from the other patients, but they said that they were nocturnal and there were 2 brothers. And you just, except for the McCarthy family, in the usual alternating hemiplegia, you don't see 2 brothers with this disease.
It's the history of the disorder is a very interesting one. My wife mentioned Simon Verrett, who was one of the pioneers of pediatric neurology in Quebec City, and who was involved with the disorder early on, and his colleague was John Steele, who eventually practiced pediatric neurology at the Sick Kids in Toronto. But John Steele was a restless person, and he worked in many places, including in Micronesia, and traveled extensively. And the first actual paper about the condition comes from the pen of John Steele. And I don't know where he is now. He is a relatively young man, so he should be available somewhere, but I haven't heard from him for several years now.
But he did come to the meeting in Rome, which was sometime in the 80s, and he wrote the introductory chapter in the monograph on alternating hemiplegia.
It was a meeting that was organized by Frederick and Eva, who is the pediatric neurologist at the Bambino Gesù, which is the outstanding pediatric hospital in Rome, in the shadow of the Vatican.